So next immunoassay technique is enzyme immunoassay. So this is called ELISA, enzyme linked immunosorbent assay is the full form of ELISA. So this enzyme immunoassay, what we use is the catalytic properties of enzymes. So we exploit the catalytic properties of enzymes. To detect and quantify immunological reactions. So enzymes like alkaline phosphatase, horseradish peroxidase, HRP, it is called HRP, horseradish peroxidase, glucose 6 dehydrogenase and beta galactosidase are the enzymes which are most commonly used as labels in enzyme immunoassay. They are used as labels in enzyme immunoassay. So ELISA is a heterogeneous enzyme immunoassay technique. ELISA is a heterogeneous enzyme immunoassay technique. ELISA is a technique that combines the specificity of antibodies with the sensitivity of enzyme assay. ELISA is a technique that combines the specificity of antibodies with the sensitivity of enzyme assay. So ELISA is used to quantify antigen concentration. There are two types of ELISA. One is sandwich ELISA and the other one is second type is competitive ELISA. So sandwich ELISA and competitive ELISA. Okay, we will see about these different types of ELISA and the principle. So sandwich ELISA, if you see sandwich, it is a type of ELISA in which the test antigen is sandwiched between a capture antibody and an enzyme labeled detection antibody. So primary antibody and secondary antibody. So in this type of sandwich ELISA, what we do is the test antigen is sandwiched between primary antibody and secondary antibody. This secondary antibody is present on enzyme. So this secondary antibody and primary antibody, they trap the test antigen. So that is why this is called sandwich ELISA. The antigen is quantified from the amount of colored product produced by the enzyme. The antigen is quantified from the amount of colored product produced by the enzyme. Now you can see here, this is a sandwich ELISA diagram showing how we can detect the antigen. Now see here in the first step, so we take a reaction well. So this reaction well is coated with the antibody. So this antibody coated well we will take, and to this we add patient serum containing antigen. So patient serum containing antigen, we add serum to this reaction well. Now this we will let the sample to react with the antibody coated in the reaction well. So what will happen? Antibody, this is antibody and this is antigen. So there is antigen antibody reaction and complex is formed. In the next step, we add a second antibody. Okay, second antibody, that is antibody which is raised in vitro, that we will add in the form of reagent. So when you add the second antibody as reagent which is linked to the enzyme, this is linked so second antibody will be added. Now this second antibody will come and bind with the antigen over here. Now in the next step enzyme labeled antibody we will add. Enzyme labeled antibody we will add. This is second antibody. You can see this is an enzyme labeled antibody. The circle is enzyme, Y shape is antibody. So when you add the second antibody goes and binds with the primary antibody which is already added over there. Okay. So now when we add the substrate, what will happen? This antibody contains enzyme. This enzyme acts on the substrate and changes the color of the converts the substrate into product, and finally the color is changed. So this color change is reading done in the spectrophotometer, and we get the quantification. We can quantify the concentration. So this is what the ELISA technique, sandwich ELISA technique. So here you can see here the first step is here. This first step is a well in the microplate with the antibody coated, and let's say a hormone we wanted to detect. This is the substance we wanted to detect. So we added the sample in the second step. The sample which contains hormone comes and binds with this antibody. Then we add another antibody. We add another antibody with the enzyme. So this will come and bind with the hormone. So as a result, what will happen? This is one, second two antibodies, and in between antigen is present. So this is called sandwich. So up is up also there is an antigen, down also there is a antibody, down and up is the antibody, in between there is an antigen. So this is sandwich like. Okay, so the second antibody is linked with the enzyme, and this enzyme acts on the, when you add the substrate, acts on the substrate and converts the substrate into a product which is colored substance. The color reaction takes place. The colored reaction is measured at the particular nanometer to quantify the concentration. So that is about sandwich ELISA. The second type of ELISA is competitive ELISA. Competitive ELISA. It is a type of ELISA in which primary antibody is coated to a solid phase and equilibrated with the standard and the test antigen So then the enzyme labeled immunogen is added, which will bind to the primary antibody at sites unoccupied by the enzyme labeled immunogen. The antibody is quantified from the amount of colored product produced by the enzyme. The antibody is quantified from the amount of colored product produced by the enzyme. See here, we will take a well which is coated with the antigen. So this is coated with the antigen. You can see this is antigen. Antigen coated well we will take, and then you add antibody containing serum sample containing antibody you can add here. So antibodies will come and bind with the antigen. Now we will wash this. Then again you add enzyme linked antibody binds to the specific antibody. So enzyme linked antibody will come and bind with the primary antibody. Then again you wash it. Now after washing you add the substrate. The substrate converted by the enzyme into a colored product. The colored product is taken. The colored product is considered and measured at the particular nanometer to measure the concentration. So this is also called as indirect ELISA or competitive ELISA. If you see the applications of the ELISA technique, so ELISA is mainly used for the determination of small quantities of proteins like hormones, antigens, antibodies and other biological substances. Now commonly used pregnancy test for detection of human chorionic gonadotropin in urine is based on ELISA. You know that we have for HCG ELISA technique is there, and we are doing fluorescent immunoassay. Earlier we have this strip method in the lab. Before starting chemiluminescent immunoassay, we have the fluorescent immunoassay machine, that is biomerics machine. Biomerics machine is there. There we are doing HCG test which is based on the this fluorescent immunoassay. Okay. Then diagnosis of AIDS test can be done by using this ELISA. Detection and estimation of the antibodies against HIV, hepatitis, toxoplasma markers, all these things can be done by using this enzyme immunoassay technique. Then That is about enzyme linked immunoassay technique. Okay.